It's on.
All right. Hi, everybody. This is the procedure that you have written in your lab on how to extract DNA from strawberries. So I have some pieces of strawberries. I'm going to put them into this baggie here. I'm going to take out as much air as possible. Okay. And I'm going to start to smush up the strawberries. Okay. So take the air out. I'm going to smush up the strawberries. So my fingers are going to break apart the large portions of the strawberries, so that they don't resemble strawberries anymore. Because our whole goal here is to try and figure out what the DNA of a strawberry looks like, okay? And if all living things have DNA. So strawberries should, from what you know, right? Strawberries, are they living? Yeah, they are living. So they will have some kind of DNA. So I've smushed up my strawberries pretty well. And then I'm going to add something that you probably have in like a spice cabinet in your house. It's called meat tenderizer. So I'm going to take this meat tenderizer, sprinkle it on my strawberry mush here I have going on. Okay, and then I'm going to smush it up again, take out all the air. Okay, and this meat tenderizer is kind of cool because what it does is it condenses all the DNA in the cell. Right, so remember where the DNA is found in the cell, right? We found our DNA in the nucleus. Okay, I know you were thinking that at home. It's going to condense the DNA in the nucleus, and it's going to break down the proteins there. So, next thing I'm going to do is I'm going to measure. This is called extraction buffer. It has dish soap and salt and some water in it. I'm going to measure 10 milliliters, like the procedure says, of extraction buffer. And I'm going to open my baggie again, very gently. And I am going to throw this extraction buffer in there. Right, so this extraction buffer, first off, the dish soap is kind of cool because the dish soap is going to break apart the fatty parts of the cell membrane that controls what moves in and out of the plant cell. Okay, the salt is going to break up the proteins that are around the DNA so we can start to make the DNA visible. So now you can see I kind of have like this slushy mixture here going on. Okay. Mix it so it's kind of like a liquidy mush here. Then I'm going to make this apparatus as your picture says in your lab. So I have a funnel. I'm going to put the funnel in the test tube. And then I'm going to cut myself a piece of this material. It's called cheesecloth. Okay, and cheesecloth, you could use something very thin like the cheesecloth, but cheesecloth is going to help us filter out the large and the small parts of the cell that are left over, because we only really want the center of the cell where the DNA is. So I'm going to put this cheesecloth in the funnel. The funnel's in the test tube, and then I'm going to very carefully, I'm going to pour this mixture into the funnel. Open it nice and wide, right? And then I'm going to make sure all my strawberry gets in that cheesecloth there. And I'm going to start to let my cheesecloth there, right? And you can see this is what the strawberry mush looks like in the funnel there, right? So I'm going to move my funnel around a little. I might actually even squeeze it a little so the juice starts to go down in the cheesecloth into the funnel, through the funnel into the test tube. So look, this is all the small part of the DNA, right? The small part of the cell with the DNA in it. Okay, so we're going to let it sit there for maybe a minute or so. All right. Here, give you guys a better view here. Cool. Great. Now we're going to take the funnel out of here. Sorry about that. And the next step says to pour some ice cold ethanol. Ethanol is what you guys know, what everybody knows as rubbing alcohol. Okay. I'm going to pour 5 milliliters of rubbing alcohol into this graduated cylinder. And then this rubbing alcohol is actually pretty cold. I put in the freezer before this. And The rubbing alcohol will start to separate the mixture from the rest of the materials in the nucleus, okay? So this mixture is pretty heavy. It's got sugar, it's got salt, it's got soap. The alcohol is actually less dense, so it's gonna sit on the top of this mixture. So I'm gonna hold a test tube kind of like sideways or angled, and I'm gonna slowly pour this alcohol on the top of it. So now you'll see I've actually started to develop 2 layers. This top layer is going to be the alcohol, your rubbing alcohol, and this pink mixture that's starting to form. So I wonder what that pink mixture is going to be. The bottom is going to be mostly the strawberry material we have left, some of the soapy water, the salt, etc. So you can see now, which is kind of cool here, the DNA has floated to the top. How cool is that? Okay. So this mixture at the top, this kind of like pinkish color, that's all the DNA from my 2 pieces of strawberry, right? Crazy. Now those 2 pieces of strawberries have millions of cells in them. So this is millions of pieces of DNA from my strawberries using just the normal stuff I have in my house. So let's refer back to this video as we need, and we will answer the questions about DNA extraction.